Hello, everybody. So today, sorry about that, I'm zooming around. We are going to talk about genetics. We're going to get into those Punnett squares that you enjoy so much. And here we go. The objectives for today are all of number 7. We'll cover each of these. And for number 8, we'll cover the monohybrid crosses today, which look like this with the 4 squares, and we'll get into those larger, more complex dihybrid crosses next time. So in the Wiser Me worksheet, take a minute and fill in the discussion. What do you think of when you hear the term genetics? And what about the term heredity? So these two are similar, but they're not exactly the same thing. So genetics is actually the science of something, just like biology is the study of life. Genetics is the science of heredity. It also includes the mechanisms by which traits are passed from parent to offspring. By mechanisms, I mean meiosis, essentially, that whole diagram of the meiosis with the gametes that then get the gametes then come together through fertilization and you end up with a zygote. All of that is included in the study of genetics. Now, what you may be thinking of is heredity specifically, and that's the passing of traits from parent to offspring. And this is more typical of that Punnett square kind of stuff that you're used to. So one thing that I like to clarify from the beginning is genes versus traits. So here's the definition of a gene, which you have seen several times now. A short segment of DNA that codes for a specific protein. So when we talk about traits, we have to talk about proteins. Proteins play all kinds of roles in your body. They are the building blocks for many of our physical traits. They are the building blocks for the melanin that colors our skin. They're the building blocks of our fingernails and our hair. But they're also the building blocks for things that we cannot see. If you remember all of those transport proteins that are located in our cell membranes, those proteins are created with instructions from the DNA. There is a gene that codes for those transport proteins. So our physical traits are things that we can see, but they're also things that we can't see. So when we talk about a trait is typically a characteristic, usually that is visible, but it doesn't have to be. So being lactose intolerant, for example, could be a trait. That's something that's genetically determined. But we're going to use examples that are easy to visualize. So we're going to talk about physical traits like coloring, blood type, hair, freckles, whether your earlobes are attached or unattached, all of that stuff. Which leads us to some more vocabulary, genotype versus phenotype. If you look at my picture over here with the DNA, our chromosomes, we've got 2 chromosomes here. It's probably a homologous pair. These chromosomes code for, these genes code for what the organism will look like. So the genotype is considered the entire genetic makeup of the organism. And then the phenotype is the physical appearance, or it could be an internal characteristic, like maybe one of your transport proteins doesn't function correctly. So that is a genetic trait that you have. It's a phenotype because even though you can't see it, that protein, that transport protein is a physical thing. It's an actual thing that's in your cells, microscopic, so you can't see it, but it is physically there. And if those transport proteins aren't functioning correctly or are not made correctly due to a genetic error, you could have a variety of different diseases. Some of you may have heard of cystic fibrosis. That's a disorder that's caused by a protein that's not made correctly. So that means the DNA code is off for that protein. It's not made correctly, and it is in the cell membrane, but it can't do its job properly. So it results in a disorder. That transport protein is still part of the phenotype, even though you can't see it. It's the physical trait. All right, so your genotype and sometimes your environment determine your phenotype. So this is overly simplified, my picture here, that your genotype codes for your phenotype. But you guys probably are aware that your environment can influence your phenotype to a certain extent as well. A great example of this is height. So you do have genes that indicate the general potential of your height, whether you'll be tall or short. But your environment, the food that you eat, the nutrition that you have, especially when you're young, will also impact how tall you are. So in that case, there's a genetic and an environmental component to that trait. So some things are purely just based on your genotype. Other things can be influenced by your environment as well. And those things together or alone, just your genotype, will determine what you look like, your phenotype. So let's talk about alleles. Based on what I've seen from you guys already, you have a pretty good grasp of this, but here's another short definition. So alternative forms that a gene that occur in the same location on homologous chromosomes. So here's a picture of homologous chromosomes. These are actually the two that line up together in prophase 1 and then go through crossing over. So you can see how if these two were homologous and lined up next to each other and they were to swap the tops, they would be swapping the gene for eye color. And so the information doesn't get messed up, lost, or duplicated. The alternative forms of a gene, there can be lots. So you actually only have 2 of these alternative forms of the gene. So let's say that eye color is ruled by just this one gene, which actually isn't true. There's lots of them, but let's just say there's one. And there are lots of different eye colors out there, right? You can only have 2, one from your mom and one from your dad. But there are lots of other ones out there. And these are just the 2 out of the whole mix that you happen to have. So how do new alleles arise? I mean, are there any new eye colors? Are there eye colors today that didn't exist hundreds of years ago? That's a good question that I actually don't know the answer to. But new alleles arise primarily through mutations. So alleles are actually the little genes. We can create new phenotypes, new traits through crossing over, because you can mix some traits with others that have never been seen before. But when it comes to actual alleles, the actual gene, the code on the DNA, that can only happen through mutation. And that can impact the phenotype of the organism. All right. So those little alleles, the mom and dad copy that you have, one of those is going to be expressed. In other words, one of those is going to be seen or show through as the actual trait. So you have 2 alleles for every trait, and these alleles interact with each other. So the one that you're most familiar with, the interaction that you're most familiar with, is the pure dominant and pure recessive. And that's really where we're going to stay today. You may be familiar with or have already encountered things like incomplete dominance and things like that. We'll save that one for next week sometime. Today, we're just going to work with pure dominant and pure recessive. And the definitions are there. If it's dominant, anytime you have that allele, it's going to show through. Recessive means that the only time that that trait is expressed or shown in the individual is when there are 2 copies of the recessive allele, so they're the same. And just so you know, I put this little note down here. I think there's only like 12 to 15 or 18 traits, human traits, that are actually pure dominant and pure recessive. The rest of them are way more complicated than that. One of those traits is whether you have attached or free earlobes. So I have a picture there of each one. You can look in the mirror. Mine are the free earlobes. So you can kind of see the background of my hair there. I actually have an earlobe. If you have attached earlobes, they just come straight in and connect without that little hanging piece like you see here. So let's talk about earlobes. All right, so these combinations of alleles, you have two copies, right? So we're going to talk about the combinations you can get from mom and dad. And I like this visual vocab picture. These are talking about peas in terms of wrinkled and round, which we're talking about earlobes. I guess you could have wrinkled and round earlobes. But so homozygous means that the two alleles are the same. I will caution you, if you use this word homozygous, you must follow it up with homozygous dominant or homozygous recessive. It's not enough just to say homozygous, because then I'm going to ask you, well, okay, they're the same, but is it the dominant allele or the recessive allele? So the other one is heterozygous, where you have two different alleles. Again, let's take a look at this word. I like the visual vocab, but we can break this down even more. You guys know that the prefix homo means same, and the prefix hetero means different. And after going through the reproduction notes, what is a zygote? A zygote is that initial single cell, that fertilized egg. So you have a fertilized egg cell, the very first. It has exactly 46 chromosomes, and it's a little zygote. So in order to have two of the same alleles, you have to be at least a zygote, right? Okay, so homozygous. Zygous is talking about that initial fertilized egg, that initial set of DNA. And that initial set of DNA gets copied and copied and copied and copied and copied through mitosis. And that's why all of the cells in your body have the same DNA. All right, Punnett Squares. Yay! I know, I'm so excited. I wish we were in class together so that we could celebrate. We're finally here. All right. Definition. I know, how exciting, right? We all know what it is. But it's a diagram that's used to determine the probability of having an offspring with a particular genotype. And from there, you can determine what the genotype. So we're going to use this very old drawing from a very old textbook, but I kind of like it. I mean, it's pretty good. So this is a body cell, that's why it's round, and the mom is pure recessive. Attached earlobes are the recessive trait. Free earlobes are the dominant trait, and this is the body cell of dad. And so when mom and dad get together and they have some offspring, the child has to get an attached allele from mom, because that's all mom has. The kid has to get an allele for free earlobes from dad, because that's all he has. So the offspring ends up being heterozygous. All right. So as you're building Punnett squares, here's some basic rules, and they are rules. You can't break them. Makes me nuts. And in some cases, it's actually wrong. Dominant alleles are always written with a capital letter. Recessive alleles are always written with a lowercase letter. When we get into things like codominance and incomplete dominance, we'll have other ways of indicating which is which. But for now, all dominant alleles are capital, all recessive are lowercase. If you are handwriting homework, or if you print out a worksheet to fill out on paper, or sometimes even if you are doing OneNote and you're using your stylus, choose letters, and it doesn't matter. You can use the letter A for every trait that we talk about. It doesn't really matter. But choose a letter that looks different in their capital and lowercase form. O is a terrible one because when you write those little O's next to that box, it could be very hard for me to tell which is the capital and which is the lowercase O, because these, it's kind of easy to tell because there's context, there's other letters around it. But if all you have is a bunch of O's in a box, it gets really hard for me to read. So that is a Miss Collette rule. Use letters that have two different forms. Some of you, your capital W's are rounded and they look just like your lowercase w's, so stay away from those. If you do like a sharp pointy W for the capital one and a little rounded one for the lowercase, then by all means use it, but just make sure they're very different. All right, capital letters are always written first. So when you're filling out your Punnett square, it doesn't matter whether it was on the top or the side, the dominant one always goes first. I also have students who have a tendency to want to call like a capital F for the free earlobes and an A for the attached, and then you end up with two different letters for the same trait, but there's always one letter per trait. So if you're talking about earlobes, you can just go with the letter E, which is what we will use. And then the capital E is dominant and the lowercase e is recessive. All right. So the first thing that we have to do when we talk about Punnett squares is actually go back to meiosis. So here we go. In meiosis, we start with the germ cell. So this occurs in the ovaries for us ladies and in the testes for the guys. You have the germ cell. And we're going to go with mom here. So mom has all recessive traits. So she has all lowercase e's. Why are there 4? There are 4 because these two on the left represent that X. So there's like a little e at the top of this one, and there's a little e at the top of this one. The 2 on the right indicate her second copy of that allele, and it's a little e on this end and a little e on this end. In the first division of meiosis, we separate those homologous pairs. And then in the second division of meiosis, we then separate the sister chromatids into the gametes. So the gametes, in this case, are either sperm cells or egg cells. And you can see down here at the bottom that if she made 4 egg cells, which we know from the last video that that's not quite right, but if there were 4, the only possibility in her egg cells is a lowercase e. The same would be true for dad because he only has the dominant trait. So there would have to be capital F's in all of the sperm cells that he would make. All right, knowing this, we are going to take possible gametes from mom and put them at the top of the Punnett square. So you can imagine like the possible eggs. And if you look up here, all of mom's eggs have only one possibility. So that means we're going to put the lowercase e's up here for earlobes. And dad, we said the same thing. He only has the dominant trait. So the only possibility for his sperm cells are the capital letters. So this is how you set it up. One parent goes on top, one parent goes on the side. It doesn't matter which one you put it in. So it doesn't matter if we were to put the dad's alleles at the top and the mom's, well, I guess they're the gametes, and mom's gametes on the side, it wouldn't matter. And then you look at, well, what would happen if this egg cell and this sperm cell got together? And you would put those in this box. So actually what we do now is we bring down this letter E into every box. So bring this one down here, and this one comes down here. And then we bring the other gametes over. So this sperm cell is going to fertilize this egg. The sperm cell is going to fertilize this egg. This one's going to fertilize an egg. This one's going to fertilize an egg as well. So these are the possible combinations of egg cells and sperm cells for these two parents. Easy peasy, right? Okay, so what do we do with that? When you look at the results, so these are the possible combinations of offspring. This does not mean that if they have 4 children, this one is done, this one is done, and then this one and this one. What this means is this is the probability for each child. Each time a sperm and egg cell get together, it has the same probability of being any of these. I say that because when you make an egg cell or a sperm cell, do you actually lose the DNA? No, you just make a copy of it. So these are copies of mom's DNA and copies of dad's DNA. It's not, mom and dad aren't actually losing their DNA. So it's not like if they have 4 children that this is children number 1, child number 2, child number 3, child number 4. This square represents every time they have a child, these are the possibilities. And in this case, all of their children will be heterozygous because that's the only option. So what does it look like when we have other things? So let's take a look at this one. We have genotypic results. We can talk about those results as a percent or a ratio. So here are some flowers. These flowers are purple. And in this case, there is a capital P, which means that purple is dominant. And there's a lowercase p. And in this case, the flowers are white when they're recessive. So dominant trait is purple, recessive trait is white. And I like this diagram because it shows the mom here and it shows egg cells. So yay, mom has a capital P and a lowercase p. So those are the possibilities that can end up in the egg cells. The father plant has sperm cells and the possibilities are a capital P and a lowercase p. So when we fill this square in, what if we join this sperm cell with this egg cell? We get this one. But what if this sperm cell happens to join with this egg cell? We would write that here. So you would fill these in by pulling this one over. These two would come over and these would come down. When you get it all filled in, it looks like this. So now we have offspring, possible offsprings. If these two sperm and egg cells get together, you have a homozygous dominant individual. If these two down here, this one and this one get together, you have a homozygous recessive offspring. The other two are heterozygous. So how do we present that in terms of ratios and percents? So the homozygous dominant, what you do, there are 4 possibilities, so it makes it easy, 25%, each square represents 25%. So homozygous dominant, there's only 1, so that's 25%. It's also a 1-to-3 ratio. So for the offspring that they have, there's a 1-to-3 ratio for homozygous dominance. The same is true for homozygous recessive. There's only 1 square here that's showing homozygous recessive, so they are also 25%, or a ratio of 1-to-3. So then we go to heterozygous. There are 2 individuals here that have the opportunity to be heterozygous, so that makes it a 50% chance that the offspring will be heterozygous. The ratio for that is 2-to-2 because there are 2 that are heterozygous and 2 that are not. Always reduce these if you can. So we're going to reduce that to, it's a 1-to-1 ratio for every heterozygous individual that you have, there's one that is likely to not be heterozygous. All right. Let's take this same example and look at the phenotype. So you'll notice on this slide, I changed the listing over here from the genotype to the phenotype. The phenotype is simply a description of what it looks like. In this case, it's purple flowers or white flowers. When we look at the phenotypic results do not always follow the genotypic results because our heterozygous individuals will show the dominant trait and not the recessive trait, which means 75% of our offspring will be purple. So that's a 3-to-1 ratio, while 25% of our flowers will be white, which is a 1-to-3 ratio. So stop and think. Again, we're just talking about pure dominant, pure recessive. Can you figure out the genotype of a parent by collecting information about the offspring? It's an interesting question. The answer is yes. So let's say you have a purple flower, but you want to know what its genotype is. How would you go about finding that information? What would you have to do? Put a response in the worksheet. Okay, so you would have to do a test cross. So you can pause the video and write some of this down later or get it out of your book. But the sheep over here is a pretty good example. So let's say this is our purple flower, or we have a dominant sheep with dark fur, and we don't know what the phenotype is. It could be homozygous dominant, or it could be heterozygous. We would have to cross it with a homozygous recessive individual. And because the sheep is white, we know that it's homozygous recessive. Then you breed the two together, and you can't just make one baby, right? You have a whole bunch because you have to end up with lots of offspring, because there are black sheep for both possibilities, right? They could still have a sheep with black fur if the dad is heterozygous. They could still have an offspring with dark fur if the dad is homozygous dominant. So you have to have a whole bunch of offspring, which is why you wouldn't do this with people. It's tempting to think about those earlobes. So mine show the dominant phenotype, but I have no idea what the genotype is. My husband happens to be homozygous recessive. He has attached earlobes. I still don't know what my genotype is because we've only got one kid. And that one kid has the dominant phenotype of free earlobes. We know that my kid is heterozygous because he got a recessive allele from his dad, but he got a dominant allele from me. But I'd have to have a lot more kids and see if any of them ended up with a recessive trait. If I had a whole bunch of kids and one of them ended up with the recessive earlobes, we would know that I was heterozygous. If my husband and I had a whole bunch of kids and they all showed the dominant phenotype, then there's a pretty good chance that I'm homozygous dominant. Because I'm not having a whole bunch of kids. So test crosses are not something that you do with people. It would be easier to go back and look at my family tree and look at my parents and see what their earlobes are like, and then look at my grandparents, and there may be that would be better, more ethical way to determine it. And so we will talk about pedigrees and family trees later in the unit. And that's it. Have a wonderful day. Email me if you have questions. It's good to see you.